Hi, my name is Caeley Bryan, and I will be presenting a paper entitled High-Fat Diet-Induced Obesity, Ablates Gastric, Vagal, Afferent, Circadian Rhythms, which was written by Kentish et al. In 2016. So for a little background on the subject, obesity affects more than 35% of all American adults and is a leading cause of death worldwide. And although you probably don't think about it much, humans and mice eat at regular intervals in accordance to circadian rhythms, which are usually regulated by hormone levels, primarily leptin and ghrelin, but they're also regulated by things called clock genes. Gastric vagal afferents are activated during digestion, and they help to regulate eating habits. So primarily this paper had two research questions. Question one was, do the changes in circadian uptake of food in high-fat diet-induced obesity correlate with disruptions in gastric vagal afferent circadian rhythms? And question two was, do high-fat diet-induced circadian changes to those gastrovagal afferents occur in conjunction with weight gain or with fat consumption? So how did they figure that out? They used 7-week-old male C57B6 mice, which were randomly assigned to either the standard laboratory diet, which is SLD, or a high-fat diet, which is HFD, with an N of about 60 mice per diet. They were on diet for 12 weeks, and then post-mortem, the stomach and esophagus were removed with vagal nerves intact and placed in Krebs solution. Mucosal stroking was performed to check the mechanosensitivity of those GVAs, and was performed again before and after application of both leptin and ghrelin to test the effects of those hormones. Tension testing was also performed to test the tension receptors, and it was performed with a lever pulley system and also performed after application of both leptin and ghrelin to test the effects of those hormones. Quantitative real-time PCR was performed on nodose ganglia to test different clock genes. Then they had a separate cohort of mice that was fed either SLD or HFD for 2 weeks instead of 12. This is because the primary weight gain that occurs due to high-fat diet occurs after 2 weeks. So this was to test whether the change in eating habits or any changes to the ganglion was due to weight increase or to fat consumption. So let's take a look at those results. There was a significant increase in body weight and 24-hour energy expenditure, and a significant decrease in 24-hour movement in high-fat diet mice versus SLD mice. Those HFD mice had a decrease in movement during the dark period only, but the energy expenditure changed significantly in both, as you can see in these graphs up here. Relative to those SLD mice, food intake during the light period increased in those HFD mice, as well as meal size. So this is the quantitative real-time PCR results. As you can kind of see in each of these graphs, the lines are very similar. There was no statistical significant change in clock genes studied in either group. So you can kind of infer that those genes are not a huge result to any circadian changes that occurred. So in SLD mice, there was circadian variation in tension receptors, but in high-fat diet mice, this was not seen. But these mechanoreceptors, not the tension receptors, had circadian variation in SLD mice with an acrophase at ZT9 with negative linear correlation to gastric content, whereas high-fat diet mice exhibited minimal variation with zero correlation to gastric content. And these are the results for leptin versus ghrelin. Leptin caused inhibition of tension receptors in HFD mice, but not in SLD mice, with zero circadian correlation. Leptin potentiated mechanoreceptors in SLD mice with an acrophase at ZT3 and a drop through the dark period, but failed to impact HFD mechanoreceptors at any time. Ghrelin caused inhibition of tension receptors regardless of diet or time phase. There was no real difference there. In HFD mice mechanoreceptors, ghrelin produced a static level of inhibition that was not seen in those SLD mice. So let's take a look at those 2-week mice. Their sensitivity of the tension and mechanoreceptors varied with circadian rhythms, which is consistent with wild-type mice, but not with the diet. So this shows that change in sensitivity of those GVAs is not due to fat consumption, but rather due to weight gain. So the real takeaway for this is that high-fat diet-induced obesity eliminates the circadian variation of GVA mechanosensitivity, and that change in GVA sensitivity is likely due to weight gain. It's not due to fat consumption. However, due to all of our results, the mechanism is still undetermined. We can't directly correlate it to gene upregulation. It's more of a behavioral shift. So there's still a lot of different things to study here. So if you have any questions, thank you for listening.